Yeah, thank you so much, Gustavo, for the introduction and for inviting me to this webinar. I'm excited to talk about this topic that we've chosen together. And what I try to do today is to present data out of our own lab and data from colleagues in the field to try to lead you through the story of the intertwined connectedness between the metabolism on one side and immune function and immune phenotype on the other side in the postpartum cow specifically. And I look forward to your questions in the end. So in the outline here, just wanted to briefly give you a rundown of what we're going to be talking about. The particular situation of the peripartum cow. You are all here likely because you know that the postpartum cow is at specific risk for diseases that have to do with the immune system, not potentially functioning properly. I want to introduce you to the concept of immune dysfunction. We're letting go of the idea of cows being immunosuppressed during this time, and I'll try to argue for why we say so. We'll focus some on inflammation because that is one of the features of the postpartum cow's immune system, that we see this heightened state of inflammatory response. And we'll cover the link epidemiologically between metabolism and inflammation. And from there, we will examine the relationship from both sides, as I introduced. And I'll end up with a summary. So briefly, for an introduction of this particular period of time, I'm sure many of you are aware of this. We're dealing with this drop in dry matter intake around the time of calving, but at the same time, cows are starting to make a lot of milk very early on in lactation, and this discrepancy between the cow's ability to eat sufficient amounts of digestible feed and her needs for nutrients that go into milk production lead to energy balance deficits during this time. And not only is there an energy balance deficit, we need to always keep in mind as well that we have a deficit in amino acids. Concurrently, our insulin concentrations drop in the cow, and there is a lack of glucose availability. At the same time, of course, we see an increase in lipolysis, usually measured by an increase in fatty acids in the bloodstream, or NEFA, an increase in ketone bodies, typically in the blood measured by BHB, or you can use urine or milk as well. We see a muscle loss that is reflected in the negative protein balance, that some of this muscle is metabolized to fill amino acid needs. And then there's also particular time for oxidative stress during this period. And of course, calcium should not be forgotten, especially when we're talking about the immune system. There's this brief period of hypocalcemia right after calving, and we will talk some about this as well. So in summary, we're dealing with a negative energy, protein, and mineral balance with the onset of lactation. And this is very simplified. There is, of course, many other things that are happening, but for the purpose of today's presentation, this is what I want to focus on. So you came here to learn a little bit more about the immune system of the postpartum cow. Recently, with two of my colleagues, we put together a review on this very topic. And here we're summarizing the functional changes in selected blood immune cells in the transition period. And I don't want to go into too much detail and bore you with kind of biology level content about these different immune cell subsets, but just broadly speaking, there are three important immune cell classes that we want to talk about: neutrophils, monocytes, and lymphocytes. And in general, you see most of their functions being decreased. So most of these arrows are pointing downwards, meaning that the function of these cells is decreased during that particular time in the postpartum period. However, not all functions are decreased. You see some of these arrows pointing upwards. For example, the production of inflammatory cytokines by monocytes and mucosal interferon production after mucosal vaccination, intranasal vaccination is increased. And you even have some evidence that IgA production on mucosal surfaces can be increased during this particular time. So not everything is globally suppressed. Why is this happening? There has been a number of really beautiful experiments that help us understand this. In this particular set of experiments, that is a summary of the papers that came out by Kimura and Noneke. They come from the same study that was performed. They tested the effect of parturition alone with or without a functional mammary gland. So in these cows that they examined, they surgically removed the mammary gland months before parturition, and then they studied changes in the physiology of these cows. And by doing so, they could pinpoint changes just due to parturition, to giving birth, or to parturition and the starting lactation. So that's what I'm depicting here in the middle. With this red cross, I'm showing you this group up here, which has a parturition, gives birth to a calf, but there is no functioning mammary gland. And here on the left, just as an example of some of the findings they had, I'll show you that the proportion of T lymphocytes in the peripheral blood cells is dropping. So in these cows that are giving birth and lactating, there is less of a proportion of these lymphocytes. Okay, whereas that is not happening in cows that are purely giving birth to a calf. On the right-hand side is serum IgG, and that's very interesting and something we should keep in mind when we talk about immunity and the immune system of the peripartum cow. There is a lot of IgG, as you can see here in these cows that are having a lactation, that is decreasing in the bloodstream and disappearing. And some of that, of course, as you know, is going into colostrum. But there's, of course, other needs as well with the IgG in the body. But big difference here in cows that give birth to a calf alone, or those that also have colostrum synthesis and then colostrum and milk production. So not everything is lost, though. I'm trying to give you this idea of letting go of the immune system of these cows is just weak. For example, here, a piece of evidence that came out in 2017, they tested the effect of vaccination intranasally in cows at different times in their production stages. And when they vaccinated cows in the fresh group, speaking here about 24 hours after calving, or 2 weeks before calving, they actually saw an increase in IgA production after intranasal vaccination in these fresh cows. So that was some of the evidence that some of us took to say, okay, the immune system is not globally suppressed and unable to cope with challenges at this time. However, as I said in the beginning, the inflammatory response, so the innate response of the animal to an inflammatory stimulus is greatly heightened. And I want to show you some data here from Julie Opgenorth. She's done a series of very nice experiments to help us understand this physiology in the periparturient cows compared to cows in mid-lactation. And here in this particular work, she compared giving cows either in early lactation or in mid-lactation a very strong stimulus, a lipopolysaccharide, which is a cell wall component of E. coli. So it's essentially telling the cow, wow, there's a bacterial infection, respond to this. And as you can see here in this graph, in early lactation, these cows respond with much greater increase in body temperature than in mid-lactation. She also found that TNF-alpha concentrations, IL-6 and IL-10, are heightened. And this work is nice because it confirms what we have long thought, that the inflammatory response of these cows is just increased in this postpartum period and not decreased. So with this short introduction, what I'm trying to tell you is that there is some decrease in immunocompetence, but that is partially so. And we have inflammation that is enhanced. So during this period of time, we have coined this transition period immune dysfunction, rather than talking about immune suppression. So I would argue that instead of having to look for a stronger immune system, we just have to look for a smarter immune system. We have to particularly look at the things that are decreased in their function and see if we can increase those, and vice versa. We have to look at the heightened inflammatory response and see if we can find ways to mitigate that. And why that is, I will introduce in the next few slides. So inflammation is the first defense to a stimulus. And you've all seen these types of pictures, the 5 cardinal signs of inflammation: heat, redness, swelling, pain, and loss of function. That is the immune system's immediate reaction to any stimulus. So if you poke yourself with a needle, you will have these 5 signs of inflammation coming in relatively quickly, right? So you all have observed that, and it is critical for quick resolution of an injury or infection. What happens during inflammation? For some of you who might not be looking at these types of cells all the time, I will just give you kind of a very broad overview. We have, of course, circulating immune cells, and we have immune cells that sit in the tissues. For example, here, immune cells that sit in the adipose tissue. Then there's endothelium and platelets involved in this response, as well as the liver plays a very important role in inflammatory response and in the immune system in general. The immune system gets activated by different stimuli. PAMs and DAMs, that's stimuli where the immune system sees certain patterns that are associated with danger or pathogens. And LPS, for example, counts in this category. Then, of course, just a simple injury is also activating the immune system and inflammation. And then necrotic tissue and dead cells, which in the postpartum cow, you can imagine the recovery from birth, the uterus that needs to shrink and resynthesize lining, has a lot of necrotic tissue and dead cells that need to get removed. What are the effects of such an inflammation or such a response? We have an increase in cytokines, an increase in all these other mediators that I will not cover in detail. But what happens now is that we get these 5 signs of inflammation, and we get production of what we call acute phase proteins. And under these, one that is getting talked about a lot is haptoglobin. And I'm sure all of you have heard about it. And because it's been such a hot topic in the transition period, I just wanted to introduce that to you a little bit more in detail. So haptoglobin is something that it's a mediator, it's a protein that gets synthesized in the liver upon insult or infection when there's an increase in circulating cytokines. The liver then redirects amino acids to building these acute phase proteins, among those being haptoglobin, one of the positive acute phase proteins. It also directs amino acids to build more of the negative acute phase proteins that decrease the inflammation. So less of these will be available. And an example here is albumin. Haptoglobin really is a transport protein. It does not have primarily a role in this relationship of the postpartum immune complex, but the sense that it has is that it makes iron unavailable to bacteria. So that's its primary purpose. And it increases unspecifically with just any sort of inflammation, trauma, and even stress conditions. And it has been in the literature found to increase after conditions such as mastitis, inflammation of the gut, or called enteritis, peritonitis, infection of the abdomen, pneumonia, inflammation of the heart lining, endocarditis, endometritis, inflammation of the lining of the uterus, fatty liver that you're all familiar with, which is often present in the postpartum cow. Parturition alone also increases haptoglobin. Starvation does. Transport can increase it, and even subacute ruminal acidosis. So you see here a whole host of conditions that can increase this haptoglobin. So it's an indirect but longer-lived marker of certain types of inflammatory responses. So just keep this in mind as we go forward. And is inflammation always bad? You can see I'm trying to home in a little bit on this inflammatory response of these cows because that's such a feature of their particular physiological situation. And I will say that a little early on is always good and actually necessary, as I will show you. And here in this slide, I have put together a comparison of 2 studies, one performed in humans on the top and one performed in cows on the bottom. And you can see that we're showing acute phase proteins in humans. Instead of haptoglobin, we measure what is called CRP, but just consider it kind of the same global marker as we use haptoglobin. And you can see here that after delivery of women giving birth to babies, the acute phase protein is slightly increasing over a period of time and then coming back to baseline about 48 hours later. In cows, you also have this increase, and this is data from Julie Hussey, where it increases slightly and then decreases again and comes back to baseline a little bit later, and that's because haptoglobin has a longer half-life than CRP. And we know that this inflammation is necessary to lead to detachment of the bovine placenta. And when we interfere with this inflammatory process before the placenta is detached, we can actually delay the detachment of the placenta. And what we then find is a retention of the placenta or retained placenta. So is inflammation always bad? No, some of it is necessary, as I just explained to you. But if we have too much and for too long, then we have negative consequences. And here in this slide, I put together, again, some of the data from Julie Hussey, where I only showed you that little insert that is marked with the red box here, but now I'm showing you a more complete picture of this work. And here she compared healthy cows, mildly metritic cows, and severely metritic cows, so cows with either a mild uterine infection or a severe uterine infection after calving. And you can see that the haptoglobin goes up much higher in these cows that have a uterine infection. And so, yes, as summarized by Dr. Bradford here in 2015, that's now 10 years ago already, and we're still wondering a little bit about this physiological situation, is that, yes, we want this increase in inflammation, but we want a rapid resolution, because then we have good metabolic adaptation and normal estrous cycle, so better reproductive success. However, if we have this heightened inflammation or this chronic inflammation, then we have this association with negative energy balance, metabolic disease, and infertility. And what are the connected points? How are we going about looking at the relationship between the immune system of the postpartum cow and metabolic status? So let's consider a few of the examples of inflammatory or immune-mediated diseases in the postpartum cow. Metritis or infection of the uterus is one of them. And here we see a loss of immunocompetence and generally an increase in host-damaging inflammation. For those of you who, like me, treat cows on farms, you know that cows can get very sick with metritis, and it's often life-threatening if they have puerperal metritis, meaning metritis that is very soon after birth. And that is because there is an increase in this host-damaging inflammation that is something that the cows are battling with at this time. Similarly with mastitis, those of you who engage in controlling mastitis know that mastitis right after calving is the most dangerous, particularly when it's a gram-negative mastitis. Again, there is loss of immunocompetence. The cow is unable to clear this infection well, and at the same time there is this increase in host-damaging inflammation. So we're seeing this heightened and more chronic inflammatory response in these cases. And the metritis is out a little bit further in this time scheme. And many of you probably work with cows and score these cows on an endometritis score. Again, it's a loss of immunocompetence. The cow is unable to clear the bacterial infection in the uterus. And here we have a chronic inflammatory response with lack of resolution postpartum. And that, of course, is known to have a negative impact on fertility. So what does the literature tell us about associations of inflammation and metabolic maladaptation postpartum? I introduced to you the specific metabolic situation that the cow is in postpartum, and I introduced to you the specific changes in immune parameters that we can find in a cow postpartum. But how do they fit together? We now know a little bit more about this. There's been some recent papers, and I summarized them for you just to give you an idea in the different categories. Here, starting with the uterus, the odds for metritis by 14 days in milk were found to be about 2 times greater for cows that had hyperketonemia postpartum, or in other words, a BHB concentration between 1.2 to 1.4 millimoles per liter. Similarly, cows with endometritis at 28 days in milk had approximately 2 times greater BHB concentrations 2 weeks prior. So here is the association between metabolic maladaptation and inflammatory disease and loss of immunocompetence. Similar results have been published for mastitis, infection or inflammation rather of the mammary gland. Here we know that the incidence is 2 to 3 times greater if BHB was over 1 millimole per liter, 1 to 3 days in milk, as shown here in this work by Giannossi. And this was particularly severe for gram-negative pathogens causing mastitis. And in another work that was published by Robertson, we see that clinical mastitis odds were 1.6 times greater if, again, cows had hyperketonemia in this time postpartum. So I hope that I can convince you that there is some association between the metabolic situation of the cow and these immune changes or immunity changes, but it's a chicken or egg question. So what comes first and who influences who? We're going to just examine this in both directions. We're going to look at metabolism and inflammation affecting each other, and we're going to start with how inflammation affects metabolism. In this work, out of Dr. Bradford's group by Yuan et al. In 2013, they tried to isolate the effect of what would inflammation do to the cow's metabolic response in the first 7 days postpartum. It's a very elegant work. They used recombinant TNF-alpha and infused it daily under the skin. And then they measured intake and metabolic effects, water intake, milk yield, and other parameters that I will not all mention today. But what they found was that these cows that were treated with TNF-alpha, which is a pro-inflammatory cytokine, which I introduced earlier, that is also responsible for increasing haptoglobin, leads to an intake suppression, negative effects on water intake, decreased milk yield and components, and of course increased the blood haptoglobin concentrations. And here in this slide, I'm just showing you the dry matter intake suppression in these cows. So here's the control that did not receive any TNF-alpha. And then in a biological response here, the more TNF-alpha these cows received, the more severe was their intake suppression. And this intake suppression, again, some really nice data from the study from Julie Opgenorth, is more severe in early lactation than it is in mid-lactation. I'll walk you through this graph. Here is the intake of 4 groups of cows before they were stimulated with this LPS, which is a danger signal to the immune system, which then will lead to inflammatory response. And these cows were either given LPS in mid-lactation, in the solid black here, or in early lactation. And each of these 2 groups also had a pair-fed group, meaning a group that was only fed as much as the group that received the LPS. But for the purpose of this, just ignore that and look at these graphs that tell you that the mid-lactation cows here jumped down in their intakes from about here down to here, but look at the decrease in dry matter intake in early lactation, which is much more severe, and they don't recover as quickly as the mid-lactation cows. So again, early lactation cows particularly prone to this heightened inflammatory response, and the intake suppressing effects in these animals are much more severe than when cows have an inflammation in mid-lactation. In this particular work, that was performed in mid-lactation and currently is the best data we have to kind of understand the potential energetic needs of inflammation, was performed in mid-lactation cows by Sarah Quidera. And in this work, they tried to isolate how much was the glucose need, so the sugar need of a cow after a single inflammatory event. And this red line here are cows that received LPS, but then had an infusion of glucose to keep their blood concentrations of glucose pretty steady, as opposed to cows that received LPS, but then the authors of this study let glucose drop as it would normally after an inflammatory response. And then there are some control cows here as well. So very nice, again, elegant work to try to understand this. Again, I want to highlight it's in mid-lactation cows that are responding differently to glucose homeostasis than fresh cows. But just to show you here how much the authors calculated a single inflammatory event would cost the cow in terms of energy needs. This is the infusion curve of glucose that they had in the study. And they had to give these cows about 265 grams of glucose to keep their blood concentration stable. And they had a large loss in milk yield as well. And when they put these 2 together, so first the glucose that they had to infuse into the bloodstream to make sure these cows wouldn't drop in their glucose concentrations, as well as the cost calculated for the milk loss, they came up with over 1 kilogram of glucose for a single event. Now, this was very isolated, just the effect of inflammation on glucose, of course, and as I will show you later, there are other effects on other metabolites and nutrients, such as amino acids, that were not taken into account here, but also have a metabolic cost. And some of our work also shows that the glucose availability at this time postpartum is already low. So that's why I'm telling you we have to be a little bit careful to interpret these findings in mid-lactation cows and translate them directly to fresh cows, because their glucose concentrations are already decreased, both in the immune cells, shown here in this red box, This is what we call the Buffy code. When you spin blood down, you can actually find these cells sitting between the red blood cells and the plasma. So we looked at the glycogen concentration in these immune cells, specifically in the transition period. And as you can see, the concentration in glycogen, which is a storage form of glucose, drops pretty solidly in this first week postpartum before it starts increasing again by 4 weeks after calving. And on the right-hand side, just a very simple curve to show you the glucose concentrations in the blood of cows. There's a little tiny peak around the time of calving, which is likely caused by the glucocorticoid response, the stress response of these cows around parturition. But then you see this large decrease in glucose concentrations in these cows in the first week postpartum. So again, there's already a dearth, a lack of glucose, so to say, in these cows. And so if we were stimulating them and measuring their glucose needs for an inflammatory response, I wonder how that would look. So that would be work that still needs to be done. Many of you work in the field, and you don't have to have these studies that infuse TNF-alpha or give LPS. You actually have your own experiments very often. You see them in terms of costs, metabolic costs of vaccination. Here are some studies. Unfortunately, we don't have a lot of good data on this, but 2 studies that I have summarized here for you that use different vaccinations in cows and generally observe a 1 to 3 kilogram drop in milk production. And that's something that all of us see in the field, especially if we're vaccinating cows for the first time with a new antigen that they have never seen before. We see a more pronounced drop in milk production even. And that's a combination of a little bit of an intake suppressing effect, but probably to a large degree as well, the metabolic effect of the activated immune system that we see here. Acute inflammation, as I said, also decreases plasma amino acids. And Tawny Chandler, former postdoc in my lab, did a very intricate study where she looked at amino acid concentrations in the blood of cows that, again, were stimulated with LPS. And you can see a trend here. It's one of our nice model systems to just make cows have this robust and predictable inflammatory response. But we were particularly interested in what would happen with amino acids in these cows. And you can see here, she split this up by non-essential amino acids in black, essential amino acids in red, and branched chain amino acids in blue. And this is in the 24 hours following relative to the IV LPS bolus. You can see that there's a drop here in these concentrations. Particularly the essential amino acids and the branched chain amino acids showed this drop before they have a rebound effect here 24 hours. The cows did not eat very much, as you can imagine, when they got this LPS and had this inflammatory response. I did introduce to you that inflammation has an intake suppressing effect that is mediated by cytokines, for example, TNF-alpha. So then we were wondering, is this decrease in amino acids just purely due to these cows not eating, or is the immune system really using some of these amino acids? And Trent Westhoff, a former PhD student in my lab, had included this question in his own PhD work, and again performed an intravenous LPS challenge and measured amino acid concentrations. And these are very colorful charts, but just to orient you, again, we're looking at 24 hours in relationship to making this cow have an inflammatory response, looking at essential amino acids on the left and non-essential amino acids on the right. And this black line is cows that did not get LPS, but also weren't allowed to eat. So here we're asking, if a cow is not eating, would her amino acid concentrations fall to the same level as cows that are not eating and having a response to LPS in terms of inflammation? And no, cows that you are not letting eat don't have this drop in amino acids. But cows, and these are all the colorful groups here, received some LPS, have this pretty sharp drop in amino acid concentrations that rebounds by about 24 hours, both for the essentials and for the non-essential amino acids. So this is giving us, again, strong evidence for the immune system actually using some of these amino acids actively. An inflammatory response also decreases blood calcium. And here, Tawny put together a graph where she compared the ionized calcium concentrations in cows that she stimulated in the first week postpartum, again with LPS. And you can see this dramatic decrease in calcium concentrations over the first 24 hours after LPS with a nadir at around 5 to 6 hours. And the nadir and calcium concentration coincides with the height of the fever response. Okay. So you can see here, cows have this very dramatic, again, fever response. If you've ever done an LPS challenge, you know these cows really go very high in their body temperature, sometimes very concerningly so. But this is the nice part of this model system. There's no live organism involved. They will start to decrease slowly and come back to baseline at 24 hours. So we asked again this question that I just introduced to you for the amino acids, wanting to know, does this calcium just decrease because cows are not eating while they're having this inflammatory response? And again, Trent picked this up and looked at this in his own IV LPS challenge. And in black, again, the cows that were not fed in the same fashion as the cows that received the LPS, but showed no decrease in calcium concentrations. And this is done at about 22 days in milk, so about three weeks in milk, while the cows that received the LPS have this sharp drop in calcium concentrations. Okay, so again, we know that the immune response alters the calcium concentration in the bloodstream and makes it less available. And you will often see these cows having these extreme inflammatory conditions such as metritis and mastitis, have cold ears, and exhibit all the signs of a milk fever. So I try to introduce to you the relationship of this chicken and egg conundrum, and we are now going to be examining it the reverse direction. So what are the metabolism effects on inflammation? And just to remind you of the metabolic situation that we're finding ourselves here with this graph that I introduced to you in the beginning. So let's start with energy balance, if we examine that effect on the immune function, immunity in the postpartum cow. The best way to look at energy balance, I believe, is to look at the cow's intake capacity or their voluntary intake. And again, very nice work by Julie Hussey here in the periparturient period in cows that are observed for their intake and then followed postpartum for about 21 days to see who of this cohort developed mild and severe metritis. And in this graph, we're looking at time here on the x-axis, of course, and the dry matter intake in kilograms on the y-axis. We are making a metritis diagnosis, obviously, only after calving has occurred. And in red are shown the cows that are going on to have a severe metritis, a severe infection of the uterus, and they already show this drop in dry matter intake before they calve. Okay, so there's already something that is different in these cows before they even go on to have their calf. And I would argue that, of course, the uterus is not inflamed at the time of these cows still carrying their calf. Similarly, in this work by Hammond in 2006 published, he focused on endometritis, and the same exact relationship was found in cows that were going to go on to endometritis. These were added in the study, and the cows that also were developing metritis had a decrease in their dry matter intake already in the 1 and 2 weeks before calving. So again, giving us the evidence that a drop in dry matter intake, meaning there's less available energy and nutrients, is actually predisposing these cows potentially for this infection of the uterus. At least it's associated with each other. So energy balance we examined. Now we're going to move on to the increase in NEFA, ketones, and oxidative stress. And a series of very nice infusion studies here singling out the effect that hyperkalemia could have on the immune system. On the left, by Zarin in Rupert Bruckmeyer's group in Switzerland, and on the right, Turner Swartz, who did this work when he was with Dr. Barry Bradford. And these fit very well together to give us an idea of what does a cow do during times of hyperkalemia, and what is that specific effect of high BHB when there is an immune challenge. On the left-hand side, we can see these cows that received the BHB in blue in both cases. both on the left and the right. I have highlighted them in blue, and then the control cows that did not receive BHB. So you kind of have to imagine these cows are hooked up to an infusion, and there's constant infusion of BHB into their bloodstream. So we're holding them at a constant level of about 1.7 to 1.8 millimoles per liter here, which we consider hyperkalemicemic. And then these cows are being challenged on the left-hand side with LPS, again on the right-hand side with actual live bacteria into the udder. And we see that on the left-hand side, one of the effects of having this high BHB at the time of immune stimulation of the udder, that there is not as many somatic cells coming into the mammary gland. So we're having a blunted effect of cells responding in the mammary gland to the stimulus. And then on the right-hand side, the cows that were infused with BHB at this intramammary challenge with strep uberis, live strep uberis, had much higher numbers of these bacteria after this challenge, meaning that the cows were less competent to clear this infection. Highlighting though that this was done in late lactation, so we would have to consider how this would look in fresh cows. And Dr. Abuelo, who's a colleague of mine who works extensively on oxidative stress, gave me these few slides that I wanted to include to you because, of course, oxidative stress is also something that has an effect during this time. And due to the decrease in dry matter intake, there's also a decrease in the antioxidant that cows are having available. But at the same time, they have this increase in the metabolic demands for antioxidants. And the result of this discrepancy is an increase in ROS production. Also, as I mentioned, there's a increased stimulation of the innate immune system in these cows. And when these cells get activated, they also produce more ROS. So altogether, this increases the production of ROS in the cow. And when we are at times of energy deficit and there is an increase in lipolysis, the NEFA concentrations by themselves can also have an effect on increase in ROS production because there's increased need for metabolizing these floating around fatty acids in the liver. And due to this increased ROS production, we're having this multiplying effect of ending up in a situation of oxidative stress. And why am I including this here? We know that the redox status or the balance between antioxidants and pro-oxidants can alter the immune response at the site of infection. And the generation of ROS is positive in a way because it is a pathogen killing mechanism of these immune cells. But if it gets too much, then there is excessive ROS production, and that can decrease the immune cell function further, even beyond what I introduced before. So in summary, he would say that oxidative stress diminishes the functional capabilities of immune cells. So going back to this diagram and looking briefly at the effects of calcium, here I have contrasted two studies, one from our lab conducted by Tony Chandler, and here on the other side, again, by Julie Opgenorth. This is very nice to be able to look at these together. On the left-hand side, we have an early lactation study, and on the right-hand side, we have a mid-lactation study, and I'll walk you through this. First, here in early lactation cows, we stimulated these cows either under the condition that their calcium concentration was held constant. So Tony would use an infusion where she would constantly measure the calcium concentration and maintain these cows at a normal blood calcium and did not allow them to drop in their blood calcium like they would normally do with inflammation. And then she measured the intravaginal temperature because that's a very reliable indicator of their core body temperature. And she found that there was no significant effect on the infusion of calcium or the provision of calcium to the immune system in these early lactation cows compared to those who did not receive calcium. A little bit of the shifting of the curve, but nothing significant. On the other hand, in Julie Opgenorth's work, she tested the effect of either giving calcium intravenously or orally. And I'll just highlight the intravenous calcium response because it translates the best to what I'm showing you on the left-hand side. And this is this top curve here. We're looking at hours relative to their LPS challenge. And you can see that the cows that received intravenous calcium at 2 time points throughout this study had higher rectal temperatures. So they did observe a response in the temperatures when they were giving these cows calcium. So I think more work needs to be done to understand, is this an effect of cows being in mid-lactation versus early lactation, or potentially just a difference between the studies being conducted? But there is some evidence potentially that providing calcium at the time of an inflammatory stimulus may exacerbate some of the inflammatory responses that we see, although we don't see strong evidence of that happening in early lactation cows, which of course are the ones that we supply calcium to the most. We would not very often supply a mid-lactation cow with calcium. And then lastly, just to mention glucose, amino acid, and insulin. In one study, not to forget about that, the amino acids play a very important role in the immune response of the postpartum cow. And here we performed a study Dr. Chandler did in the early parts of her postdoc, where she infused cows for 4 days with amino acids to essentially take away the negative protein balance that fresh cows would normally experience. So as you know, cows drop in this energy balance and amino acid balance, and she supplied them with this human-grade infusion product that is meant to be used in pediatric medicine. Many of these bags went to these cows to try to obliterate this protein deficit entirely in the first week postpartum. And then she asked, what would happen if I challenge these cows now and make them inflammatory after they received amino acids before? And we saw some changes in their inflammatory cytokine responses. TNF-alpha was slightly decreased here, and we also had less of the anti-inflammatory response of IL-10. And together we took this to mean that the cows responded less with an inflammatory response than the cows that did not receive the amino acids, which would fit with the existing literature when you isolate cells and treat them with amino acids. However, interestingly, when we did that, the cows also had an increased intravaginal temperature, and this was quite substantially different in these cows, but by 0.6 degrees Celsius in their maximum response. So while the cows did not make as many cytokines as the cows that had not received amino acids, they had a more robust inflammatory response. And then we tried this out, and we wanted to say, what if we actually feed cows? Is this something practical where we can alter the inflammatory response of postpartum cows by feeding them more metabolizable protein? And that again was picked up by Trent Westhoff in his work, and he had a feeding trial with 92 transition cows. And although we had very different treatments as far as the protein balance goes in these cows, the MP balance, he did not observe an inflammatory response. The cows just made more milk by giving them the amino acids. So in the postpartum cow, it's very difficult, and we have seen this in several projects, very difficult to direct nutrients to where we want them to be, because the cow is overriding most of what we want her to use it for with the need for milk production, because that is her priority in a homoerotic response. So in summary, I hope I could show you with this compilation of the data that we have available that there are certain changes in the immune system that are driven by lactation, that are directly influenced by the cow preparing to make milk and making milk, as I showed you in the beginning. There are some early inflammatory responses that are physiologically and required postpartum, and we should not interfere with them because it has negative consequences. However, heightened and extended inflammation costs unnecessary nutrients, as you've seen, costs us milk production, and of course, that is a financial cost to the farm. I want to highlight that the nutrient requirements that you have for inflammatory responses are on top of all the other requirements that the cow already has at this very fragile period of time postpartum, and that the effects of the early inflammatory process and intake suppression are long-lasting. So if we curb the lactation curve of the cow in these early days, then this will have long-lasting effects on the entire lactation of the cow. And as you've seen the effects on the uterus with the data I showed you on metritis and endometritis, we also have long-lasting effects on reproductive success. Likewise, we believe that early intervention or prevention of this heightened response of inflammation can show long-lasting consequences. And some of that could be due to making sure that we don't see these decreases in the intake due to the intake suppressing effects of inflammation mediated by the cytokines. Immune response and metabolism are intricately linked. If you ask me who is driving who in this relationship, I would not be able to tell you that it's a one-sided relationship. I believe that we have to consider both in this specific situation. However, I have to say the cost of inflammation on metabolism is out of our control. If a cow responds with inflammation, she will use up nutrients just for this inflammatory response that are no longer available to her for other purposes. So therefore, we have to prevent unnecessary metabolic effects that decrease the immune competence. So making sure that these cows are not in severe negative energy balance, are not lacking protein, are not having a decrease in calcium concentrations that may have intake effects, are not having excessive oxidative stress, etc. And at the same time, we need to prevent unnecessary exacerbation of inflammation and its associated metabolic costs. So preventing metritis, preventing mastitis, and having these inflammatory diseases reduced will also reduce the metabolic costs to these cows. If you came here today to find a silver bullet to understand transition cow immunity and fix it, unfortunately, I have to tell you that there is none available to us today unless somebody hasn't told me about it. It's very complicated to try to direct nutrients, as I said, in the postpartum cow because she will override anything that we give her and purpose it to make more milk. Therefore, it goes back to the principles that we have known about for a long time. We have overall transition cow management being likely much more important than trying to supplement individual nutrients, making sure the intake is adequate, making sure rumen health is on par, and decreasing disease risk with everything that we have available for preventive medicine. None of this is really truly my hands-on work, and so I want to highlight my lab, former and current members that do all of the hard work and that have contributed to our understanding of this, as well as the many colleagues who work in this field. And I think together we're trying to move forward in understanding this better and hopefully identifying some of the key points where we see some way of intervening in these relationships. I do have my email address here. I think we have plenty of time for questions, but if there's something after this webinar that has piqued your interest or that you disagree with and you want to share with me or want to start a discussion, please reach out to me by email. And Gustavo, I hope that we have some questions in the chat that we can go over. So thank you very much for your attention.